This session will be about MLC, and it's my pleasure to give the introduction, and I'll just briefly go over some general information so that I'm sure that everybody is on the same page. So for some it might be something they know already. This is my funding, it causes no conflict of interest. We are holding a patent for guanabenzene vanishing white matter, both in the EU and the USA. We have an orphan designation for it. I'm a consultant for Calico on therapy development for vanishing white matter, and I'm a co-investigator for the Alexander disease trial of Ionis. This is the program for today. So I'll start with an introduction. Then Christina will talk about progress on Alliance MLC. Then Rogier, sitting here, will talk about what happens in the MLC brain. Then Quinty, also sitting here, will talk about why is volume regulation defective in MLC. Then Jigyasha from India will talk about the Indian experience. Then Marla Chaplot will talk about abilities in MLC. And then we have a final discussion. But I think also in between we can have discussion. If anybody has questions, that's fine for me. So to begin with, it's important to realize that there are two forms of MLC. One is classic, and classic means that's the most common form of MLC. Then there's the remitting form of MLC, which is much rarer. Let's start with the classic MLC. Patients develop a big head in the first year of life. You can see that here, that this is the upper limit of normal, that head size rapidly increases, and that most patients are way above to standard deviation. Initially, development is typically normal or a little bit delayed. Many patients walk a little bit later than healthy children. And then from infancy on, there can also be occasional epileptic seizures. And status epilepticus is, considering the mildness of the epilepsy, relatively frequent. Occasionally, and I must say many patients have not experienced that, there's a temporary decline after head trauma, and it's characterized by seizures, sometimes even prolonged coma, but very different from vanishing white matter. Typically, patients return to what they were afterwards, so there's no permanent effect. After a couple of years, a slow motor decline starts, mostly coordination problems, and most patients become wheelchair dependent as a teenager. There's a late and mild cognitive decline, especially slowing, and behavioral and psychiatric problems may occur. There's a variability in severity. I know of 2 patients who never walked. I know also of patients who still walk in their 40s and sometimes have a business. Inheritance. With the more genes we find it becomes more complicated. So most often it's autosomal recessive. That means that both parents are carrier of a gene mutation, and that a patient is a double carrier and has nothing to compensate it with and has the disease. That's how it works. And sometimes it's dominant. That means that it is inherited from the father or the mother right to the child. And but it may, if it's dominant, it may also be de novo. So there may be a new gene mutation occurred in the child. Laboratory tests in MLC reveal nothing. Then we have the MRI. When you have a disease, it's always important to realize what defines a diagnosis. Often it's a genetic test. That's not what defines the disease in MLC. In MLC, it's the MRI. We have still patients in whom we cannot find a genetic defect, but the MRI is classic for MLC, so they still have MLC. It starts like this. This is normal. You can see here the vertical image, or T1 weighted image. You can see the normal white matter is white and the gray matter is gray. These are horizontal images, and you can see the normal white matter is dark gray and the gray matter is light gray. What you can see in MLC is that the color of the white matter is all wrong. It's dark here on the vertical image and it's all white on the horizontal image. You can also see that the white matter is swollen. If you look at the folding of the brain, you can see that the foldings are broader than they normally are. So there's swelling of the white matter. You can also see that here. But the gray matter looks fine. Over time, this is an MRI of the same patient. You can see that the swelling remains. The cyst in this patient have become more and bigger. That's not always the case. And then this is an older patient, 13 years old. And you can see that the swelling decreases over time. So here is the brain is filling the whole skull. Here it's already a little bit room on the outside. Here it becomes more. And this is a patient in her 40s. And you can see that there's now clear-cut atrophy. Still the white matter looks a little bit swollen with broadening of gyri. But the ventricles have become big and the cerebral mantle has become much thinner. The subcortical cysts are always there. Yeah? If anybody has a question, if I'm unclear, let me know. So what's important to realize is that this patient of 6.5 years was clinically normal, whereas this just had a big head, whereas this patient is wheelchair dependent. That this patient is normal with such an horrendous looking MRI means that it cannot be demyelination. Radiologists typically tend to report these MRI as demyelination, which is all wrong, because if this would be a fully demyelinated brain, the patient would be completely devastated and wheelchair dependent. So you know from MRI that that cannot be true. It's something else. The MRI actually, we knew that from the beginning when we defined the disease in the 90s, that it looks like there's too much water in it and it's just swollen. Like the structure is pretty normal because the child is doing well, but there's just way too much water in it. Brain edema. Feel free to take a picture. Yeah, no, because I think it's informative. With diffusion weighted imaging, you can look at the freedom of water movements. So if the structure of the brain is really dense, then the water have very little movement, freedom of movement. So, and we express that as apparent diffusion coefficient. The apparent diffusion coefficient normally is around, let's say, 90. In MLC, you can see that the apparent diffusion coefficient is way higher, is in the 200. And that means that there's a highly increased water content and the water spaces are large. So there's much freedom of movement. We did, in my first patient in the 90s, a brain biopsy. When we were clueless. We had no idea what it was. What we see in the subcortical white matter is that there are countless vacuoles filled with water. But you can also see this is a myelin stain. The myelin is dark purple. The myelin is there, confirming it's not a demyelinating disease. Then the question was, where are the vacuoles? This is a stain for myelin also, and you can see that the vacuoles are covered in brown, meaning that they are covered by myelin. We did electron microscopy, and what you can then see is this is a vacuole attached to a myelin sheet. And this is the covering of the vacuole with very high magnification, and then you can see dense lines and a double intraperiod lines, and then again a dense line, a single dense line. There's no membrane in the body that has that apart from myelin, so that must be myelin. And then you can see the last thing. This is a blood vessel. This is an endothelial cell. This is a red blood cell. And then you can see an astrocytic end feet. And the astrocytic end feet is also swollen and also contains vacuoles. Now, what about astrocytic? Okay, so there are fluid-filled vacuoles with a myelin sheet, and astrocytic end feet are swollen and also contain vacuoles. Myelin, we have now two words, myelin and astrocytes. So these are nerve cells in the gray matter. They have axons, and they are covered with insulating material that we call myelin, that mostly helps with efficient impulse conduction, and this myelin is produced by oligodendrocytes. In MLC, there are vacuoles in the myelin. Then the green cells that are now covered by text are astrocytes. Astrocytes have end feet on sort of everything. on blood vessels, on nerve cells, on myelin, and they are the housekeeping cells. So they are central in brain homeostasis. They make that everybody is happy and healthy, that you have your bread when you go to school, and that you're dressed well, and that your hair is combed. That's what astrocytes do. And so they are central in iron and water homeostasis and in volume regulation. And we will have a next talk going what it is about. Okay. And can I have my that are forwards? Can I? Okay. Oh, okay. Then the improving, the remitting type of MLC. You can see here the normal again, and this is a child of 9 months with a big head. You can see it's just the same. The white matter is swollen. There's an anterior temporal cyst, so MLC. But then at 8 years, the disease is gone. You can see it's just normal. Even the anterior temporal cyst is gone. And when I first saw this, I thought there was a mix-up of MRIs, and I asked for the right one, but it was just the same patient. So what you see, what happens is it starts out typical, then you can see the improvement here, and then it ends that the disease is gone away on MRI. Clinically, these patients also have a macrocephaly, big head in the first year of life. They have also a mildly delayed of normal development. Epilepsy may occur, but is less often than in the classic phenotype. And then from the second or third year on, there's clinical stabilization. The decline doesn't happen. And approximately 50% of the patients have a persistent big head, but in some patients it slowly becomes normal. And then some patients are just normal, and some patients have a mental retardation, autism, or psychiatric features. Some have a parent with a normal head size, but when you look at old pictures, you can see that this mother had a big head and she was small. In some families, the parent still has a macrocephaly, but is otherwise normal, indicating, in this case, dominant inheritance. Genetics, it's complicated. 90% of the patients with classic MLC have a recessive mutation in the gene MLC1. Approximately 10% have a recessive mutation in the gene gliocam. A few patients have a de novo dominant mutation in a new gene, GBRC5B. That's just, it's going to be published in August, but it's already online. We just found a new gene, and all patients have a new mutation, and a single one. Then the genetics of remitting MLC, almost all patients have a dominant variant in gliocam. A few patients, that's also news, have a recessive variant in aquaporin 4. So it's really complicated, and the question is whether there are still more genes. We know of at least one gene to go, so and then I think we are virtually done. Can you make it obey me again? Okay. So the conclusion is that MLC is a disease characterized by brain white matter edema, currently 4 associated genes known. And then, of course, you know, we are about therapy development. What should we do? So most patients have an MLC1 mutation, but we still don't know what MLC1 does. Although we know it now for 20 years, we know what the processes are about, but it's still something that we really don't know. So can we do gene therapy? Because we know the genes. And I think we are not there yet. I'm a little bit hesitant about the potential side effects of gene therapy versus the benefits, because MLC, to be honest, is a relatively mild leukodystrophy. It's a very slow disease, and if you lose your child because of a side effect of gene therapy, that would just be terrible. Stem cell therapy. I personally have concerns regarding feasibility and efficacy. Then drug therapy. we have just found a new gene, and I hope that Rogier is going to mention it, but a GPRC5B might be a druggable target. So that's news. Then there's a second thing, that loss of MLC1 activates interleukin-induced inflammatory signals. So could you use an interleukin-1 receptor inhibitor, anakinra, for instance? And we have been talking with the group from Rome, and we'll do a study on the subject in mice to see whether that's really effective. So currently, there's no therapy ready for B-testic now, but there are new directions. Clinical trials are critical for evaluating new treatments, but actually we are really not ready for trials. And if you come to think that we might be there in 5 years, 10 years, then we really need to become trial ready now, because becoming trial ready takes years. What's needed for trial readiness? Insights into the natural disease course. Validated outcome measures, clinical measures, body fluid biomarkers, MRI. So these are necessary for the design of an effective and efficient clinical trial. If you don't know how the disease course exactly is, you don't know what you should do. If you don't know what's a good outcome measure, you also don't know what you should do. So what I said, getting trial ready takes years. Means to achieve clinical trial readiness. I think all groups need to be organized. Patients and families need to be organized. Doctors need to be organized. And there needs to be a registry. I think those are the 3 critical things. And I mentioned them here. International MLC registry. We formed an MLC consortium. I'll just of clinical experts. I'll just show you. But and we discussed already how we will start. The basis of the MLC registry will be the Amsterdam database. We already have data on approximately 230 patients. This database will use Castor, a cloud-based secure system that can capture and integrate data from any source. But importantly, it's not meant to be an Amsterdam registry. It's meant to be an international registry governed by the MLC consortium, who will decide on things, and also with input from families and Alliance MLC. What's needed is, we are all very busy. And if you ask very busy people to do something, it takes loads of time, way too much time. So we decided on to hire a dedicated person to set up and run the registry. The necessary budget for it for 4 years, so it's approximately €100,000 per year. In Amsterdam we have already 200,000 together and probably 300,000 already available, and we aim to start in January 2024. This is the consortium. So we aim to have 7 clinicians, and it's not about research, it's about trial readiness. So we are building on clinicians with clinical and research expertise in leukodystrophy, in particular MLC, and we chose them for geographic representation. It's still a limited representation, but as you see, if you take the east coast of the USA and you go up all the way to India, it's already a time difference of 10.5 hours, which is sort of the limit you can do for video meetings. One of my PhD students is Eline Hamilton. She did a PhD and she did the first sort of natural history study on MLC, and she helps us. And the mission is mostly to form a network of clinical collaborators on MLC and achieve clinical trial readiness. So here are us. Amsterdam, then Dr. Jigyasha Sinha. She's sitting here representing the biggest MLC community worldwide, because actually MLC is very rare, but not in India. Then Pinar Tobologlu from Turkey. In the Middle East, MLC is relatively frequent. Then Francesco Nicita He is in Rome, and in Rome they have a strong laboratory MLC group with Dr. Ambrosini. So I think that's important to have. Then the East Coast of North America is represented by Dr. Geneviève Bernard from Montreal. She's sitting over there. And Dr. Ali Fatemi. And for the first time ever, he's missing a ULF meeting because he's always there. We are contemplating the 7th member, and we'll come up with it. What I think is very important is that we have an interaction between clinics, families, and basic research, and that it works both ways to achieve treatment. And this is our setup in Amsterdam. You can see we have a laboratory, and we also have multiple clinical leukodystrophy experts. So I think that's how it should be. And this is my group in Amsterdam. I'm not sure, this might be the last slide. I think so, yes. And but what I should stress as a last point is that the collaboration with families is extremely important. We can set up a registry, but if people don't fill in the questionnaires and forms and provide information, we will be nowhere. So it's really what people sometimes tend to think is that it's the doctors need to work and we can wait until the doctors come up with something and then we'll be involved. That's really not how it works. We need to do it together, and if there's no togetherness, there will be no therapy. Anybody with questions? No?
I'm Christina Maruda. I'm part of the Alliance MLC group, which is a patients advocacy group. I want to thank ULF for allowing us to have this session. I want to thank Dr. Van den App and her team for being partners in all of this and leading us, showing us the way to get to the next level. And of course, everyone, all the families that have joined us. We have a record number of families that have joined this year from around the world. So welcome and thank you. So I will go through what you see right now on this slide. I want to give you an update on what we've been up to since last year, when here at ULF we mentioned that we were starting a group. So during the year, we have managed to do quite a few things. We have a website, which is live, and I'll talk to you about that a little bit more. We, as Dr. Van den App mentioned, there is a consortium, an MLC medical consortium already in place. We're starting an international registry. We are planning to do the first MLC scientific symposium next year, as well as a family conference in India. And then there are ways that you can all be involved. So our website, it's alliancemlc.org. I'm not sure whether all of you have been able to look at it. I want to quickly show it to you. There is it's really the first website that has as much information that we know about MLC. And so I invite everyone to spend some time with it, especially the newly diagnosed families. We all wish we had this tool when our kids were diagnosed. So there is an about section where you can see who we are, what we're working on, also who our ambassadors are, and you can be in touch with them. The group that we have, and some of them are there, like Alexandra Chaplow is on our board, as well as even Sai, and they're there. Marla and Ron Chaplow are also there at the conference, and they've been part of our team for a long time. We have an advisory board. We have country ambassadors. So here you can be in touch with your country's ambassador. And if there is no ambassador for your country, you can be in touch with us. We have a number of partners already in place. We have started receiving donations. and of course, we need your help. There is an MLC section, which the team in Amsterdam, Dr. Min and Dr. Van den App has helped us put together. It really describes what MLC is, the symptoms, the causes. You can read about it. And then we have a research section with an overview of how the research has started and where it is right now. We have tried to make it easy for everyone to read and understand. There's also a glossary with some explanations of certain terms that are hard to understand. And then we have the research. We've spent quite some time talking to the group of our community. They've helped us put this website together, and they are hold on, can you, the internet is not working very well, but anyway, so they the researchers are in different parts of the world: Italy, Spain, of course Amsterdam, Japan, China. Here you can read about all the researchers and the work they have been doing. And you can see here the map of where the spread that MLC research has, which for many of us when we started off didn't really know. So that's the research section. Families can go and read about the patient's registry, which I will talk more about. That we have put together helpful resources, especially in the US, India, Turkey, and Japan, in those languages. So you can download those guides and have more information about your own country and how you can help the MLC affected people in your family. And then some families have also shared with us their story, and you're welcome to do that. There are ways to get involved, which I will talk about, and then we have news. So that's our website. So I invite all of you to visit it. The Medical Consortium, as Dr. Van den App mentioned, includes 6 doctors right now. Dr. Van den App from the Netherlands, Dr. Bernard from Canada, Dr. Fatemi from USA, Dr. Nichita from Italy, Dr. Sinha from India, and then Dr. Topaloglu from Turkey. One of the very first projects we will all together be working on, and by all I mean the families, the researchers, and the clinicians, is to start a registry. That's the very first step before we get into trials. And Dr. Van den App spoke about it. So what is a registry? It's a secure database of patients' data. You don't have to worry about sharing your data, because it is secure and it's in the hands of the people that need it in order for us to be able to find a treatment. Well, not us, but the doctors. It's necessary to move to critical trials, as it already mentioned. It was mentioned. We're aiming to start in early 24 in Amsterdam. It will be, of course, accessible to all the doctors and researchers around the world. It is a 400,000 euros project. 225 has been raised with, of course, the help of Dr. Van den App, with a grant from the Netherlands. So we need to raise 175,000 euros, and that's really on us, the families, and we all have to come together to make that happen. It's absolutely critical, and there are ways that we can do that. We can help you, help us. One also of the very first projects we have, because in speaking to all the researchers and physicians around the world that are working on MLC, we really realize that what is very much needed is for everyone to get together under the same roof and talk about what it is that they're working on, and then discuss in the end how we can all move forward. And so this symposium, which is the very first MLC Scientific Symposium, will take place on February 22nd and 23rd of 2024 in Amsterdam. Everyone will be there to present their projects and discuss the ways forward. We actually have guests from 11 countries, Canada, China, France, India, Italy, Japan, Korea, Netherlands, Spain, Turkey, and USA, which is incredible. And I must say that the MLC scientific community is extremely generous and ready to help us, the families. And so we're very, very grateful to everyone for their time and for making it to this symposium. Here you could see it's the map I showed earlier of where research is at, going from Europe all the way to Asia. And after we start the registry and we have the symposium, our two next important projects are going to be organizing a family conference in India and a family conference in Turkey. Those are the two countries with the most cases in the world. We plan to work with our ambassadors and the teams where we have started putting together in both India and Turkey. And of course, people from nearby, from other countries nearby are welcome to attend, but those will be conferences for families, not for doctors. So for us, the families. Sorry. Now, how can you, and by you I mean our families, can be involved? We have a Facebook group called MLC Families. It's a private group. We discuss a lot of matters that concern us parents or sometimes the actual individuals who are MLC affected. So I invite all of you to join this group. We also have a page of our organization, Alliance MLC, that's also on Facebook. So how can you be involved? You can join this group, join the dialogue, or listen to the dialogue if you don't feel that you have something to say right now. You can make a donation, and we explain on how to do that on our website. You can also be in touch with us. You can host a fundraiser. It could be a Facebook fundraiser. It could be a fundraiser in your city, in your village, in your community, and we have experience in how to do that, so we can help you. You can help us by putting us in touch with other families. It's a year ago, our Facebook group had 30 participants. Now we're over 100. The team and the family is growing, and unless we know how to reach the families, there's no way for us to help them, and there's no way for them to participate in the registry. Therefore, we cannot move forward with clinical trials and then get to the treatment. So it's all connected, it's all related. You know a family that is affected by MLC, please refer them to us via our website, via our ambassador in your country, via our Facebook. There are ways now to reach us. You can become your country's ambassador, and we can tell you what the website also includes information on what that involves. It's really being an ambassador of our Alliance MLC, helping us connect with other families, helping us help them with information, explain to them why the registry is important, and help them fundraise with us. If you have any special skills, or even if they're not special, we need them. We can only do so much. We can use help with administrative tasks, with marketing, with fundraising. There are many ways to help us. So please reach out. We have a form on the website under contact us. And there you can put the ways in which you would like to be involved. And finally, and the most important part is, as soon as a registry is ready, we will announce it, and we ask you to join it. We ask you to take the time to answer the questionnaire that will be available to all of us. We can help you also answer it. Our ambassadors will be there to help you. The doctors in your country will be there to help you. It's really critical that we all participate in the registry. And that's all I had to say. You know, it's our slogan is we fight for the day MLC is cured, and together we can do this. The information on how to reach us is here, info@alliancemlc.org, and our website is alliancemlc.org. Thank you, everyone.
So in my presentation, I would like to give a little bit of an overview of the kind of research that we're doing to understand what is happening in the MLC brain. And so first of all, my name is Rogier Min. I'm a researcher working at the Amsterdam Leukodystrophy with Marjo van der Knaap and a lot of other people there. And I'm a biologist by training, so I studied biology and then went on to study neuroscience and went on recording electrical activity in the brain and have become interested in MLC and in the kinds of things that happen in MLC for the last few years. As Mario already extensively introduced, of course, MLC is a disease with an early onset, which is characterized by a very large head in the patients, also by often epileptic seizures, which from a standpoint of understanding brain electricity is very important for us to understand what's going on. And of course, very importantly, at this point, there is no treatment at all. And that makes us in the research really focused on trying to understand what's happening, what the disease mechanisms are, and trying to find hooks where we can start to think about what we could do to restore things to normal. In this presentation, I'm going to sort of go over the genetics of MLC a little bit more in depth than what Mario just did, mainly because we have recently, as Mario also mentioned, identified 2 new genes. And I think these 2 new genes for us are very exciting because they give a very new perspective on what is going on in MLC. So I'm going to go to very basic genetics. So of course all our cells in our body contain chromosomes which contain DNA, and in that DNA we find all the genes that make up all the information that we need to generate the building blocks of our cells that make up the body. And so if we look at an individual piece of DNA that we call a gene, normally it gets translated into a piece of RNA, and the RNA then gets translated into a protein, which is a structural unit that can do something in a cell. Now, Mario already went over the 2 types of MLC that we have, and these are important to know because they are linked to slightly different defects in genes. And so if we take the first form of MLC, the classical form of MLC that was first described, as Mario also already mentioned, this is most often caused by a genetic defect in the gene MLC1. Now, the type of defect can be very diverse, and at the moment we are actually working in Amsterdam to make sort of an overview of all the different defects in MLC1 that are known at this time point. And so you can see that there are very many different sort of writing errors in the gene that make up MLC1 that can lead to a defect in the gene, and in turn to a defect in the MLC1 protein that can lead to disease. And then there is this form of MLC where patients start getting improvements on their MRI, which is very often linked to a defect in the second gene, which was discovered in 2011, called gliocam. And here again, you can see in this overview all the mutations of the gliocam gene that are known at this point. So you see there are quite a bit less than what is known in MLC1. And here you see some of the mutations are there in black, and some of them are in blue. The blue ones are the ones which are linked to the improving form of MLC, the remitting form of MLC. So if you have one version of such a blue writing error, you will get the remitting form of MLC. The black ones are the ones where you need to have 2 defects, and if you have 2 defects, you get the classical form of MLC. Oh, there's some drawing on the screen. That's interesting. Well, that's fine. Yeah, somebody does it. Yeah. So if we look at the overview of all MLC patients known to date, you can see about 3/4 of the patients have mutations in MLC1, recessive mutations in MLC1. Then about 7% have recessive mutations in GLIOCAM. So these 7% plus 76% are the ones that would have the classical MLC. And then there are these 15% of patients with the dominant GLIOCAM mutations who have the remitting form of MLC. But you see this little red piece of the pie, and this little red piece of the pie are patients in which a genetic defect was not found until a couple of years ago. And a lot of these patients ended up in the Amsterdam database, and DNA data was sent to Amsterdam. And Mario has spent a lot of time trying to find out whether we can identify new genes that can cause MLC. Sorry. And but before we go into that, I'm gonna dive a little bit into the 2 genes that we knew so far. So the genes were called MLC1 and GLIOCAM, and so they contain the building plan for the proteins, which are almost the same name, MLC1 and GLIOCAM. And here you can see what kind of structures these proteins make. So we know from looking at the proteins that they sit in the membrane, so the outer part of the cell. This one goes through the membrane for about 8 times, and it has these pieces inside and outside. And GLIOCAM goes through the membrane once, and then it has these 2 big domains on the outside with which it can latch on to other proteins from other cells. Now, looking at these structures is a way for us to try and understand what the proteins are doing. And often we do that by looking at whether they're similar to other proteins that we already know, whether they're similar to other proteins which function we already know, so that we have an idea what the function of these is. For MLC1 and GLIOCAM, that has been very hard. So we don't really find that they look very much like other proteins, and so therefore we don't really know what they do. What we do know is that the types of processes that are involved in MLC are a regulation of ions and water in the brain and a regulation of the volume of cells in the brain. Now, last year at the ULF meeting, I gave a presentation where I really went into talking about electricity in the brain, how electricity in the brain works, and why regulation of volume is so important. So I'm not going to do that again. If anybody wants to re-watch that presentation, you can follow the link over here, and you can see it back on YouTube, because all the ULF presentations are keep on being available on YouTube. And so that's more of a sort of physiological background of the processes involved in MLC. But just very briefly, as Mario also mentioned, there is a problem with volume regulation in MLC. Now, what you see here is a cell from the body of a healthy person, and this is a blood cell, and these particular types of blood cells actually have a lot of MLC1. If you give these cells an osmotic shock, so you make the amount of water on the outside of the cell higher, these cells swell up. And that's what you see here. So the cells swell up, they become much larger, and then in the period of time that follows, they actually recover their volume to their original size. And this process, actually, all of the cells in our body are able to undergo that process. It's very important to prevent damage to cells. If we do the same experiment in the same kind of cell, but now from an MLC patient, you can see that there is disruption in the ability of the cell to regulate its volume. It's not that the cell is completely unable to do it, because that would be devastating, but it is reduced in its ability to recover its volume. And so that's why we think the process of volume regulation is so important. So we call this process regulatory volume decrease, and in MLC patient cells, and also later on we found that in animal models for MLC, looking at the brain cells, this process is a little bit defective. That is giving us at least a handle on what is going on in the disease, because we know the process regulatory volume decrease, and we know that what it requires is a lot of different channels and pumps which are sitting on the outside of the cell, which allow, for example, salts and other little compounds to be shuttled into the cell and out of the cell. And so one particular player in this process has been the volume-regulated anion channel. It's a complicated name. We call it FRAC as an abbreviation. This is a little channel that sits in the membrane of the cell and that automatically opens when the cell swells up so that stuff can flow out of the cell, water can follow along with it, and the cell shrinks back to its original volume. Now, in the next presentation, Quinty, who is sitting here, is going to talk a little bit more about the types of research we are doing to understand why volume regulation is defective in MLC, because we hope that that gives us more ideas of how we might be able to target that process. But for the remainder of my talk, I would like to now focus on this little red pie here, because that also gave us some new ideas of what's going on in MLC. So, oh, this is not looking good. So as I mentioned, there were these patients who did not have mutations in MLC1 or GlycoChem, which were sort of a mystery because we had no genetic cause. And I think in 2016, Mario identified 2 new MLC-causing genes in these patients. Now, these are super rare. So they were actually 3 patients worldwide who have a defect in a gene that is called GPRC5B. And these patients had dominant variants, meaning they only had 1 copy of the gene that was altered. And these were all de novo alterations. So the parents did not have that copy of the gene. It arose in a new form in the patients. And these 3 patients have classical MLC, and that's why now the genetics is becoming a little bit more complicated, because now we have classical MLC due to 1 gene defect, whereas previously it was always due to 2 gene defects. And at the same time, there were 2 siblings discovered who had recessive variants, so on both copies of their gene in a gene called aquaporin-4. And these patients now had a disease which looked very much like the remitting form of MLC, where here you can see the MRI at a young age, which looks very much like classical MLC, and here you can see at a later age that, especially here, the white matter is now again dark colored, whereas here it was this typical light color that we see in MLC patients. And what is so interesting to us is that these are actually 2 genes that we do know of what they are doing. So we know exactly what the roles of these 2 genes are, at least in the membrane. And the second one that you just saw, aquaporin-4, the name already implies a little bit that it has something to do with water. It is actually a water channel. So it's a channel that sits in membranes of cells, and it doesn't allow, normally these channels allow salts to flow through, so ions to flow through, but this one in particular allows water to flow through. And actually, people have been studying aquaporin-4 and other aquaporins already for 20 years. They play a very important role, for example, in the kidney, but also in the brain. And in the brain, aquaporin-4, so this one is very abundant, and its location in the brain is here in these end feet of the astrocytes, the place where we also know that MLC1 and GlycoChem are sitting. So that makes a lot of sense. So we have now a water channel as a new player in the disease. The other one, GPRC5B, we actually know what kind of protein it is. It's a G protein coupled receptor, and this is a large family of receptors that are present in cells throughout our body. There's hundreds of different types of G protein coupled receptors. They all look quite similar in their shape in the protein level, so they all sit in the membrane and have several pieces with which they cross the membrane. But they can they are crucial receptors sensing all kinds of signals from the outside. So some of them sense particular molecules that come, some sense particular ions, some sense the swelling of a cell or the shrinking of a cell. And what it is that GPRC5B senses, we don't know yet. So that's something we have to figure out. What we do know is that if we look at cells from one of these GPRC5B patients with classical MLC, they show exactly the same problem with volume regulation as what we saw in cells from other MLC patients. So if the cell swells, normally it recovers, but from a GPRC5B patient, we see an impaired recovery of swelling. And the reason that we are so excited about especially GPRC5B is that in medicine, actually a lot of the drugs that are currently FDA approved, about a third at this moment of all drugs, so here you can see the amount of drugs that are FDA approved, about a third of these actually target one of these G protein coupled receptors, so one of these members of this large family of receptors. And so we're hoping that now that we know that GPRC5B is somehow involved in the processes that lead to volume regulation, maybe we can target it. And so for us, in the coming years, a big challenge will be to understand what, for example, activates this receptor, what is the consequence of activation, and can we find small molecules that target the receptor and that can influence the processes that it's involved in. So to conclude, most often MLC is caused by defects in MLC1 or gliocam. We know that patient brain cells have problems with their volume regulation, although we don't know what MLC1 and gliocam do. And we are moving forward in understanding what is happening in these brain cells, and so that's where Quinty will take on. And now we have identified defects in 2 new genes, aquaporin-4 and GPRC5B, that can also cause MLC in rare cases. And we know what these proteins do, and we hope that if we understand them well, maybe we have a new therapeutic target for MLC. So with this, I would like to end my presentation. I would like to thank all the PhD students in our group that I have been working on MLC for the past few years. Of course, Mario, Eline, Mariana, and Truus, and Huib Mansvelder, who is a professor in electrophysiology at our university that we collaborate a lot with. And I would give a big thank you to all the MLC patients and their families. Yeah, that's a great question. So for the people online, the question is, what are the kinds of stimuli that cause a cell to swell? What actually we think mainly in the brain is causing cells to swell is the fact that there is neuronal activity per se. Neuronal activity, electrical activity in the brain leads to small changes in ion concentration. So a little bit of sodium, for example, flows into the cell to cause an electrical signal. A little bit of potassium flows out of the cell to recover the electrical signal. And that kinds of movements lead to changes in these concentrations, which need to be compensated to make sure that electrical activity can be maintained. Now, what happens often is that particular types of cells, the astrocytes that we think are the problem in MLC, they take up potassium from the extracellular space to sort of restore normal situations, and that uptake causes them to swell a little bit. And when they do that, they need to recover their volume. So electrical activity per se does that. Now, electrical activity on bigger levels, such as a seizure, would do that manifold. And at the same time, head trauma, we know from experiments that head trauma can also lead to a large rise in extracellular potassium. And when cells would want to compensate for that and take up that potassium, they would again start to swell. So it's these kinds of things indeed. They are all sort of intertwined with the electrical activity.
Hi everyone, thanks for being here. First of all, I want to thank the ULF for letting me present my research here. I am Quinty. I am part of the Amsterdam Leukodystrophy Center with Mario as the head of the department and Rogier Min as my direct supervisor. So today I'm going to present my project on really trying to figure out why volume regulation in MLC and astrocytes is not working properly, and especially I will focus at the cytoskeleton. So Rogier and Mario already talked a bit about this, so I will give you a brief recap on MLC, or megalencephalic leukoencephalopathy with subcortical cysts. So it is characterized by a swollen and abnormal white matter. See if the, yes. motor problems and cognitive disability. There's no treatment, as you know, and it's caused by mutations in MLC1 or glaucom. On a cellular level in astrocytes and also in the myelin, you can find swollen astrocyte end feed and water retention in the myelin. So Rogier talked already a bit about volume regulation and the involvement of astrocytes in the brain and why they're so important. But right now we're really trying to figure out why is volume regulation not working. And we think that the problem here is the cytoskeleton in these astrocytes. So what is exactly the cytoskeleton? Yeah. So could you imagine if your body would lose all its bones? You would basically turn in a big pile of blubber, and that's not very nice. So this is the same for each individual cell in your body. They need the cytoskeleton. It's basically the muscle and the bone of the cell, and it can contract, expand, and rearrange. As you see in this little cartoon here, this is how it looks like in some of the cells. And yeah, it's basically the bones of your cell. So it has many functions. It's important for cell integrity, like it gives shape and structure. These are the highways for transport of different stuff through the cells, like molecules or organelles or other things. It's involved in cell division and cell movement, and it's also important for muscle contraction. So, like I said, the cytoskeleton gives structure and support to cells. There are 3 different kinds. So here you can see like a general how a cell looks like. So you have the cell nucleus with the DNA, and the cell membrane, and the cytoplasm where everything is like floating around. So first in red you can see actin filaments. These are one type of the cytoskeleton. These are everywhere in the cell and provide structure to these cells, and are important for mobility of cells. And for example, in your muscle cells, actin is needed to contract your cells, and so contract the muscle itself. Second, we have the microtubules in green. These also give support and structure to the cells, are needed for cell division. And for example, these are like the big, nice highways that can be used by the cell to like transport stuff through the cell. You have like these little proteins that walk on these microtubules to the cell to bring stuff to other places. And then in purple, we have the intermediate filaments. You can also know them, for example, like GFAP. That's also an intermediate filament. And these are more permanent, non-dynamic structures. So they give like this shape. They give shape to the cell. They form like this safety net to keep everything in place, basically. So for my research, I'm currently mostly interested in trying to look at the actin. So why are we interested in the cytoskeleton anyways? So my project is to really understand why defective glaucom or MLC1 in astrocytes leads to problems with the volume regulation. And our hypothesis here is that astrocyte integrity and rigidity is disrupted in MLC, which leads to defects in volume regulation. So on the right, you can see here, for example, glaucom and MLC1. That in turn influences the cytoskeleton, and the cytoskeleton is needed to make the ion channels work, which are needed again at the end for the volume regulation. And then what would happen in MLC? First, you have a problem here, so defective glaucom on MLC1. That means that the signal to the actin is disturbed, the actin is disturbed, and then the ion channels are disturbed. And then you have like the whole flow through here. So that's basically what we think is happening. So one of the experiments, one of the first things we tried, and this is in collaboration with Dr. Elena Ambrosini in Rome, we basically looked at the amount of the cytoskeletal proteins that are present in these cells. Is there any differences there? And we found a difference here. We found that if you compare healthy astrocytes and MLC astrocytes, there is more actin presence in MLC astrocytes. So this already shows you that the cytoskeleton is potentially different in MLC astrocytes. And then for a second experiment, this is I did it together with Emma Paschier, another PhD student in our group, and this is a collaboration with Nelda Antonovetta from Optics 11 Live, which is a company that provided the instrument to do this. And as I told you before, the cytoskeleton is important for the rigidity and structure of the cell. So you could imagine if you take that away, the cell becomes more soft. So with this experiment, what we basically did, you can see it on the left here, we poked astrocytes. And you could imagine if you poke something very soft, like a pillow, you can push in very, very more deeply and easily than, for example, a brick. So we did that in these astrocytes. And then you can see the results on the right. You have the healthy astrocytes in blue and the MLC in red. And we see that there is an increase in softness in the MLC ones, the MLC astrocytes. So the MLC astrocytes can be poked more deeply than the healthy ones, which means they're softer. So this is a softening effect that we found. And we think then that is also related to the cytoskeleton, because the cytoskeleton is, like I said, important for having the rigidity. So we think with this you can already see a bit that the cytoskeleton is broken down a bit there. We tried to confirm it, and for that we used an actin disruptor. This is a kind of drug that basically breaks down the actin in the cell. And as you would imagine, if you do that in a healthy cell, those cells become softer because you take the structure away. This is also what we found. So on the left, you have again the healthy astrocytes. They have like this number that means something for how stiff or soft they are. And if you put the drug on there, you can see that there's a softening effect there to the level of the MLC astrocytes. And again, if you perform this on the MLC astrocytes, you can see that there is no additional effect, which means they're already soft. It's already a bit broken down. So like making it worse, it's not going to do anything because it's already like that. So this really showed that the cytoskeleton is a bit broken, which means that those cells are softer because of that. So this is the second experiment that showed that the cytoskeleton is different and affected in MLC astrocytes. So for the last part, it's of course always nice to really visualize what you're looking at. So what we're trying to do here is to really to see what the cytoskeleton looks like in these cells, and you can do that with stainings. Here are some nice examples of some astrocytes. These are mature astrocytes, and you can see in both pictures on left and right the F actin that is stained in red and in gold. So these are some examples. So here's an example on the left of a healthy astrocyte and on the right of an MLC astrocyte. I would like to remind you that these are not like as fancy as the ones you saw before because they're a bit of immature astrocytes. That's why they look a bit different. But in red, you can see the actin. In blue, you can see the nucleus where the DNA is. And on the right, it is the same. So the next step after this, and by the way, this is in collaboration with Amélie Vreal, also in Amsterdam, was to quantify these images. And we have some preliminary data on that. So we did 2 types of quantifications. We looked at like what the area is of the actin that we see in these cells and how much there's present. And for the area, if you compare healthy and MLC astrocytes, we did not find a significant difference. So that basically means that the area and also like how big the cells are is generally the same between the 2 different conditions. This also is important that because we found a difference in actin in the previous experiment, that is not explained by MLC astrocytes being larger than the healthy astrocytes. But then we found something else that was a bit unexpected. We found no significant difference in the total actin amount that is present in healthy and MLC astrocytes. We are still working on this data set. It's quite fresh from like 3 days ago. So we're still trying to figure out what is exactly happening here. So I hope, in the next few months, there will be more on that. So for my conclusion, so if we compare healthy astrocytes to MLC astrocytes, if you look at the cytoskeleton, we can see that there are different amounts of actin present. Also, we see that the MLC ones are softer than the healthy ones. And As a third point, there's a potentially altered cytoskeleton organization in MLC astrocytes, but that is work that is still going on. So the conclusion in this is, yeah, it's still kind of a question mark, but we think it probably, the cytoskeleton is the cause that is leading to astrocyte dysfunction in MLC, but of course we need some more evidence for that. But these experiments really indicate like a first step towards that, towards that being evolved. So for the future, we need to investigate these cytoskeletal changes further, and most importantly, if we can fix them. Because you could imagine, especially for the poking experiment, it will be nice if we can use some kind of drug to get the cytoskeleton rigid again and make the cells less soft. But that is something we really have to figure out in the future. First I want to thank, of course, the family and the patients, and the rest of the Amsterdam Leukodystrophy Center, my supervisors, other collaborators and colleagues. I want to thank you for your attention, and if there are any questions, I think we'll answer them later. No questions from the audience? Yeah.
Sorry for the glitch. So I have no disclosures to be made whatsoever. So to begin with, as I mentioned that, we are seeing a lot of leukodystrophies in the country, it being one of the largest populated country, 130 million is our population. And there's this study which was published by All India Institute of Medical Science where they confirmed about 80 leukodystrophies, and we saw about 30 to 40% of our cases being contributed by MLC itself. So this is the commonest leukodystrophy that we are seeing. Now we already know from the early 1990s till date that we started with looking at cases where they had a typical case presentation with large head, developmental delay, ataxia, etc. And thereafter we saw it prevalent in certain communities of Jews, Turkish, and the Agarwal community in India. We could come up with the pattern changes in MRI brain given by Mario and the team, and identification of disease was done. We further on moved to the evolution of genetics, and understanding the natural course of the disease is still going on. And in the management, we have only the symptomatic care to offer at the moment, and we are not yet ready for the therapeutic part also. So to begin with, the country is actually pretty huge. We have these zones divided, and if we look at the zone, we have the northern, the southern, the central, eastern, western, and the northeastern part of the country. Now, Agarwals typically belong to this northwestern part of the country, and that actually constitutes the certain states like Haryana, Rajasthan, and Maharashtra. They were originally belonging to this particular part of the country, but now they are spread all over. Now, if we look at the population density of the country, even though there are some areas which are smaller, but they are very heavily populated. The eastern part of the country is very heavily populated, and also the Agarwal community is quite concentrated in the eastern part of the country. So if we look at this map, it shows how about 12 to 14 countries of the world can be accommodated in just one country of India. I mean, it has such a large, population. So why Indian perspective? Every person I meet in any meeting, they ask me, what is the incidence probably, or what are the number of cases that you are seeing? Incidence is still largely unknown. We do not have a proper data or study of MLC incidence in the country. As a clinician, I would say we are about 2,000 neurologists, about 200 pediatric neurologists in the country. Me individually has seen about 20 plus cases of MLC. So you can just imagine that the numbers must be in thousands, but we still don't know. The majority of the MLC is seen in the Agarwal community, and so now the cases which are reported in India are either the Agarwal cases or the non-Agarwal cases. And of course, there is no consanguinity as such in the Agarwal community. However, they marry in the community itself. So that is in the caste marriages. And so in turn, it probably is consanguineous. And we have a lot of bordering nations, the Indian subcontinent like Bangladesh, Bhutan, Myanmar, Nepal, and Pakistan. And a lot of patients come from all these bordering nations also. And I also feel that the ethnicity, per se in all these nations are somewhat similar. So even the Agarwals are also spread all over these nations. So to begin with the history of MLC, while in India, Professor Singhal, he mentioned seeing about 18 of these MLC cases in one of the Asian Oceanic meetings in the early 90s. It was Mario who could actually label the disease, and that's where the disease began. So it began mostly in India also at the same time. The scientists all over the world were noticing the cases in the early 90s. And this picture is very close to my heart. On the left stands Professor Bhim Sen Singhal, who is from India, who started the MLC drive in India. And on the right is Mario. And there's one study published by Leukodystrophy's Indian Scenario, where Professor Singhal again points out how MLC is the commonest leukodystrophy in India. And the mutation has also been identified, which I'll be talking about subsequently. Now this we have already heard about in the previous talks. However, what I want to point out is this is a very beautiful gene review, which is published and updated time and again by Mario and Rocker here. And this is where we get all the gene data. So we've already learned about the genes in the previous talks. So understanding the genetics in the Indian scenario, we have already learned about two different MLC phenotypes, the classic and the remitting type. And we have learned about the MLC1 and the glial-cam mutations and the remitting MLC dominant glial-cam mutation also. In India, the classic MLC phenotype is the commonest in the Agarwal community. In fact, I have not seen a single non-MLC1 gene yet identified in India. So I have also spoken to my colleagues, but they have none of them have found any glial-cam or the newer mutations yet. So this is only MLC1 which we are seeing in the country at the moment. So a little bit about the Agarwal community. So they are about 10 million in the country. And they were from the northern and central and western India. However, they originated from a village in a state of India called Haryana. And the village was called Agroha, actually. During the Mughal and the British rule of India, these Agarwals basically are the business clan. So because the administration of the Britishers were in the eastern part of the country, which is Calcutta, a lot of them migrated to the eastern part of the country. So they are quite concentrated even in the east part of the country. They are mainly the business clan. They have a lot of industry, shops, etc. And there is about 18 subcastes in the community, and they are called Gotras. Now, each of these subcastes are supposed to marry amongst the same Gotras. So that is how, the endogamy is there. They are mostly vegetarian, and they don't even eat egg, no meat, no egg. They have sort of a sedentary lifestyle, low physical activity, and high dietary calorie fat intake is there. Consanguinity is not common, as I mentioned, but the in-Gotras marriage is very prevalent. And one thing which I want to point out is that they have these big associations, societal associations like the Akhil Bharti Agarwal Sammelan, where all the Agarwals can, communicate to each other, and they have different functions and social organizations which has been established since 1983. Now, if we look at the Agarwal population, as I mentioned, the community was actually founded about 5,000 years ago. And this is a picture way back in the early 20th century of how they would dress up. And these were the 18, subcastes which I have been mentioning. So they have these different surnames. So irrespective of what surname they have, they are all in the Agarwal community, be it Bansal, Goel, Singhal, Mittal, or Agarwal also. And here we see that even though they originated in this part of the country, their population is quite significant in the north, northern, a little bit towards the western part, and the eastern, the eastern part, and the east, especially in the West Bengal region. There has also been population structure studies of the Agarwal community per se, where they have shown how endogamy has influenced, that in-caste marriages have influenced the gene evolution as such. So this is a very interesting study again. And also the genetic diseases which are prevalent in the Agarwal community, we see these are the genes, the genetic diseases. Spinocerebellar ataxia type 12 is very commonly seen, especially we are seeing one of the founder mutations has been identified again in the east part of the country. Limb girdle muscular dystrophy LGMD type 2A, where 2 founder mutations have been identified. MLC1, we have found 1 founder mutation. Interestingly, all these founder mutations, they are seeing that they are on the chromosome 22 usually. And also some of the cardiovascular diseases are very prevalent in the community. However, it is given to the lifestyle of the people also. Now, something about MLC genetics in the Agarwal community. So there was this study published by Gorospe and Singhal et al. And they found that the MLC gene in 31 patients of Agarwal community were all tested positive for homozygous insertion of cytosine in exon 2, suggesting a founder effect. So basically, they were all biallelic for the same pathogenic variant. And this was the founder effect which was mentioned by the scientists. The question remains that even though the gene has been same, I have seen a lot of phenotypic variation. There is one boy who is 24-year-old, well educated, can do his daily routine activities. On the other spectrum, I have a 5-year-old child who is already wheelchair bound. So, there is a lot of phenotypic variation despite the gene is completely same. So we need to study that in detail. There are other communities in India also where we are seeing this quite prevalent. One is the Kayastha community, Kurmi, and the Eastern India Muslim community, where they are mostly the fishermen. They are called the Mandals or Haldars. So something about how the clinical system in India works. So we have actually a referral-based practice also. From the peripheries and villages, patients are being referred to the cities actually. However, there's one interesting thing that there is no waiting period to see an expert, to see a neurologist or a pediatric neurologist. So it's about 1 to 2 weeks. If you want to see a neurologist, you can just go and get an appointment quite easily. However, the doctor is overwhelmed with a large number of patients and there is no time to dedicate to one particular disease. The next point is that after this, the patient undergoes specific tests. The good thing is that the tests compared to the world are quite cost effective. There is no waiting again for the tests. There is difficulties in the peripheries in the villages. However, there is no medical insurance in the country. So people have to pay from their pockets. So again, it becomes difficult to get the tests done. In the treatment options, we have all the types of therapies and symptomatic care available, but there is non-uniformity. You go from one center to the other, there will be different, probably not that uniform therapy will be available. The equipments, orthosis, helmets, etc., we do not have institutional supports for this. The patient will have to look for it on their own. There's no dedicated leukodystrophy or MLC center as such, and there are no clinical trials or studies at the moment going on in the country. So the challenges for the families are, one, difficult trajectory to reach the expert at times, even though there is availability, but there's lack of awareness. Affordability remains a concern. And there are no support groups or family groups till date in the country. However, Alliance MLC India is on our mind, and we are setting it up soon. And there is also some social practices, like shying away from genetics. Once you tell them that, this probably is a genetic disease, they would first thing they would ask is there a cure for the disease? You tell them there is not a cure, and they probably would not want to go for the genetic testing also. And of course, it's also patriarchy. There is difficulties, when they want to blame the spouse for the disease also. So those are some of the challenges. For us also, there's a lot of challenge, we have difficult to get people's time or dedicated family members to work on this. There's less interest if therapeutics is not involved, and they are not ready to take the charge. And we've been telling them that unless you do this, we cannot go ahead for the therapeutics, but there's always, barriers for that. And we have people who have difficulty with the technology and long distances, and also they look for faster solution. And also sometimes they have, because the availability of the doctors are so easy, that there's a general distrust, and doctor shopping also goes on. However, there is strength in India that the case burden is very high. We have to contribute to the research. And even though there is no family group, but there's a joint family concept, so there is cohesiveness in the families. The pediatric neurologists can connect to each other on a large registry and foundation. And we are seeing gradually, we are working on testing facilities and how to bring down the cost of it. And of course, there's no long waiting to reach the expert if awareness is there. So way forward is probably to spread awareness. There's web-based MLC media. We are setting up, of course, the Alliance MLC India. And apart from that, we have to team up with the organizations globally. The family meetings have to be held more frequently. And we are ready to give them clinical assistance. We are trying to set up a MLC clinic soon to start with, and formulate a national registry. And these are my goals for the next 1 year, that Alliance MLC India has to come up in India very soon. The MLC clinic has to start. The national registry should be established, at least to begin with, and start fundraising. So to summarize, MLC is the commonest leukodystrophy in India. The disease is prevalent in the business clan of India called the Agarwal community, which has likely a common genetic mutation being involved. And the case studies have shown that Agarwals are biallelic, can be helpful in contributing to the registry. Establishing Alliance MLC and holding a dedicated MLC clinic is the need of the hour, and India should be the largest contributor to therapeutic trials in times to come, I'm sure. Those are my references, and thank you very much. These pictures are all very close to my heart. That's Mario and Nicole from Amsterdam. That's my mentor from India, Dr. Rishikesh Kumar, and this is my whole team from India, the neurology team. Thank you very much.
Thank you, Jigyasha. That was very insightful. You mentioned that you're setting up an MLC clinic, and will you then also provide something that's not regular in India, like that they can consult a physical therapist, that they can have splints or special shoes made for them and things like that?
Yeah, very interesting, because it's a multidisciplinary approach and we cannot work alone. It's a teamwork. So yes, I have people already on board who can come up with the therapeutic therapies and all. So we have a rehabilitation team now set up also, and we will be working on the equipments, orthosis and helmets, so that it's easily available in the institution itself. So that is definitely on my mind. I hope so. I hope it can at least make some difference, at least to begin with. And I also have very high ambitions of having these kind of clinics more, at least in zone-wise, like the northern, central, eastern, and southern part of the country also. Yeah. Yes, sir.
One question. So, having the opportunity to follow this this, I would say, isolated gene effect. You confirm that there's a wide spectrum of severity, even though with the same mutation. So can you give some detail on which is the most severe and less severe phenotype? I mean, do you have any sort of phenotypes, or you have a certain range of severity in the phenotypes of patients that are affected and are in some way segregating the same homozygous mutation?
So very interesting. This is what we are now going to look into, the phenotypic. We have to really segregate the data properly, because we have not really put them into, the outcome scales, and to, properly give you on a scientific level that, these are the less severe or these are the more severe ones. This is just an observation in the clinic that we are seeing a lot of, having exactly same mutation, but the phenotypic variation is very, very prevalent. Right? It's very significant, notably significant, but we are yet to have a good data, and that is my next, probably project on the mind. Yeah. Yes, yes, yes. Even in the same family, I would say we'll have to really look into that. There is like a sister who has just a large head and some intellectual disability, but the brother probably has a very significant spectrum. So, yeah. Thank you very much. Yeah.
One more question. I think it's also a question to Alliance MLC, and that's not something that can be answered now, but as the biggest community is in India, how can we help Indian MLC, Alliance MLC in India, and how can we help patients? Because you can say there's no cure, but there's also a lack of symptomatic treatment in India, which is a whole different matter, because that's what we have. And I think that's something that we, that Alliance MLC may also want to consider. Yeah. Okay, thank you so much, Jigyasha.
Thank you. And my name is Marla Chaplot, as you were told. I am the mother of Aidan Chaplot, that's out in the back. He's 25 years old. And also Liam and Alexandra Chaplot, who are in the audience, and the wife of my husband, Ron Chaplot. And I am going to be speaking to you on the abilities that we can focus on, all right, rather than the disabilities. When we do get that diagnosis that somebody in your family has MLC, it's always such a shock, and the amount of emotions that you go through is, fear, you're scared, you're angry, you feel hopeless. And though it's important to allow yourself to go through all of these emotions, it's also very important to allow yourself to get over those emotions. Those are all focusing on the negatives, and it can consume not only you, but your entire family. So, the old adage of the glass half full, or is it half empty? So how do you see it? The half empty believers can actually make you feel just that. It can make you feel empty, which leads to low self-worth, low self-esteem. It could lead to mental health disorders, depression, anxiety, obsessive-compulsive disorder. On the other half of that spectrum, you have the half-full believers. Those are the ones that have the optimistic attitudes. And it's been proven that in these positive thinkings can actually increase your lifespan. It can lower your rates of those mental health disorders I just spoke of. And it actually improves your immune system, which ultimately will have less illnesses that you'll have to contend to. Probably the most important part is it can allow you to have better coping skills. We all have periods of stress in our lives, whether it's MLC related or not. We do deal with stress. Some wise words of wisdom that have been imparted to me is, if you are not happy with something, then change it. Not everything can be changed. If it is something that is unable to be changed, then learn to accept it. This is easier said than done. However, with practice and with coping strategies and using tips like mindfulness, it can be done. Mindfulness is the act of being present in the moment and accepting things as they are. And if you learn to be mindful, you'll find it easier to accept the things you cannot change. So let's focus on the positives. Thinking positively may not change the course of MLC. Let's be realistic. However, it will improve your day-to-day mood, the environment in your house, the overall health, as I mentioned, less illnesses to contend with, and your coping strategies. So let's focus on what we can do rather than what we cannot do. We're all unique. Every family situation is different. What I will be describing is what works for our family. So treat MLC, or people with MLC, as you would treat others. And it's easy for you and for others to feel empathy, to, coddle, to protect. That's what we want to do. But is it really healthy for the person with MLC? Not doing them any favors. So instead, give them chores and tasks around the house. And, we did these things for Aiden, and Aiden can thank his siblings for that, because when I had household chores for everybody to do, the everybody was Liam and Alex. And they started to sit back going, hey, how come Aiden doesn't have to do anything? So I put the unbreakable dishware that I did get, put it in instead of a higher cupboard that most people have, I put it in a very low cupboard pull-out drawer. So Aiden can then safely take dishes from the cupboard and set the table, from the dishwasher, put them away, etc. He had other tasks as well, putting toys on the floor into a little bin. I actually had a toilet bowl brush that he can clean the toilet on his hands and knees. So what that does is it gives purpose, so that even people that have a disability have a purpose, right? So you're not focusing on the disability. We grew up in a family that there was no disability in our household. And it also helps take away any negative feelings and thoughts of favoritism that might have been held. We also tried to focus on ways in our house that we could improve both gross motor and fine motor skills. We were told that the stronger you are in these areas, the more that the disease will have to take away. So keeping that in mind, if you keep exercising to the point of exhaustion, thinking we're going to go that far, you could actually be detrimental to the body. So there is a fine line. So safe activities that can be done without any equipment is we did a lot of bear crawling and crab walking. We had races because we are a very competitive family. And, we get the Twister game out, and, that was always kind of fun, and it does work on all those sorts of skills. And everybody seems to love playing video games, so we made sure a lot of the interactive video games were done on the floor. And table tennis and things like that are a good way that you could focus on working your core, because you're having to be on the floor, and it's a fun way to get exercise, but you just can't call it exercise. And for the fine motor help, playing with large Lego pieces, and it's called Duplo. They're very large, so it's easier for everyone to hold on to, and you can make creations with that, and it's great for the hand-eye, it's great for fine motor. And we did a lot of Play-Doh games. We even made our own Play-Doh, and it's really great to improve that finger strength. And it's great for everybody, not just the ones that have MLC in your family. Everyone can get involved and have fun. Again, just going to keep reiterating, instead of saying he or she can't, say why not, because many activities can be adapted. That picture didn't come out too clear, but basically, those are card holders I was trying to show. And our family loves playing card games and board games. And that's such a great way to get everybody involved and to spend time together. In fact, Aiden turned 25 last month. And one thing that we did to celebrate it is we went to a board game cafe and spent hours in there playing a new board game that we had learned. And it was fabulous. We had a lot of fun. We even went back the next day to replay the game. So, the games are a wonderful way to keep the brain active. And though it can be a challenge with fine motor skills when they do diminish, that's where these card holders come into play. You can place them in front, and then when Aiden can't reach exactly what card, he just tells us it's the third one in, etc. So yeah, it's a way to still be active. These are ways that Aiden has learned to adapt on his own. He loves playing with the video controller. You can see that on the left. And we just one day noticed that he was using his chin, and that was something he used on his own to develop to be able to use that controller. And then on the same thing, his iPad, he uses his nose to fine motor skill that because his fingers' dexterity was diminishing. And we use a lot of the use of technology. You know, you see the Alexa up there, the Google. Artificial intelligence, though it can be, you know, it's a love-hate relationship for many of us. It has been very helpful in our house. Aiden can just, you know, roll into a room and say, you know, turn on the light, turn on the TV, play music. And it's just amazing. These options are limitless for him. He can ask it to put on Netflix and watch his show. So it's been wonderful. And now Aiden has been able to set himself up with a pole, and it's eye gaze technology. And we just were set up with a communication technology clinic that assessed Aiden, and he was able to get set up with that. He's able to text and phone and play games and communicate all with using his eyes. And I think that's fabulous. But enough about the computers and the games. Let's move on to the possibilities that get us out of the house. So it's important to enroll in activities. It gets you out of the house, it's great for social interaction, and it does get your mind off MLC. Some things that are great for everybody: swimming lessons. Not only is it a great life skill, but it is also a bonus fabulous for the muscles. Music lessons, you know, whether it be voice or piano, drums, it's great for the brain. The brain has been noticed, well, music has been known to be great for the brain and also for those fine motor skills. Also, we put Aiden in Cub Scouts. Very good for social interactions. It helps improve that self-esteem. They do a lot of outdoor activities. They do like camping. You learn a lot of great life skills. A lot of these are available at very minimal or no cost, so important to understand as well. Many activities are adaptable. If you haven't already been bowling, there will be some bowling, I believe, this week. And how bowling can be adapted is with the use of a ramp. And you can just change the angle of the ramp as to where you want your ball to go. And, you know, everybody can bowl that way. Biking can be done. This is a picture of Aiden and Alex biking solo, and he's very well strapped in. It gets the legs moving. It's wonderful, and you can get those bikes new, used. There's always somebody selling one. And then we also have a dual bike. There's Aiden and Liam biking. It allows two people to pedal at their own rate. And what's really good is it has a battery, because when you have the weight of two people, Liam says he doesn't use the battery. Well, I use the battery when I go with Aiden. Especially if you're going up a steep hill, that could be quite a challenge. We also love downhill skiing, and I was pleased to learn that it is an option for people to go that require a wheelchair. So this is Aiden in a little toboggan type of thing that is attached with skis, and that is myself letting him go down the hill with that. So what's nice with adaptive skiing, it can be done where somebody can just ski on their own if they are very well apt on the upper body. And it could also be done where they have no control whatsoever. But in our case, Aiden can control the toboggan by leaning left and right to steer as he slaloms down the hill. Probably one of Aiden's favorite activities is wheelchair floor hockey. And there are people of all ages and abilities that can play this sport. You don't have to be a very well-equipped person to do so. You can be stick attached if you don't have a lot of arm mobility. So it's basically a really great way to connect with other people as well. And Easter Seals is one example of a camp that is available in North America. I don't know about other countries, but there might be camps that are very similar to that as well. And it's a camp for individuals that have disabilities. It is one of Aiden's most favorite activities. The reason Aiden was not here last year is because Easter Seals was happening at the same week. He chose Easter Seals over the ULF, so. He has met friends there that he connects with all year long. In fact, they have a countdown from the last day they see each other at camp to the following year. So it's incredible. This camp is incredible at taking the dis out of the disability. They have them doing some wall climbing, crafts, swimming. They have a big talent show at the end and a banquet. And these kids have so much fun, and adults. We do live in a very remote area, so a lot of these activities are not around the corner for us. And when we first started to get ourselves out and about, we didn't have a power wheelchair floor hockey in our town, but we joined Special Olympics chapters, and they did have swimming and bowling and track and field, lots of activities that we can participate in. And then when we did learn about the wheelchair events like the skiing and the floor hockey, we started to do the traveling, and just seeing Aiden's joy that he does have, that big smile on his face, it's worth the drive to have him participate. One of Aiden's pet peeves is when people see the chair and don't look past that. So they will talk to the person who's with him and ask questions about him, his name, his age, etc., etc. And they believe that the person in the chair cannot think for or speak for themselves. So, despite the many challenges that people will see, I just wanted to share that Aiden was able to get a certificate from college in accounting essentials. So, despite challenges that are there for anybody and everybody, I wanted to share that everybody has unique situations, etc., etc. What is your dream and what is your goal? Because you can search for it. Just remember that anything is possible. Thank you.
Anybody who wants to ask Marla something? Or okay. Yeah. Yeah. Okay. The question is a 5.5-year-old boy who has a good cognition but has balance problems. What can we do for him? I think that's for you.
For balance problems, I mean, that's a good one. I guess we did a lot of therapy or exercises that would be on the floor rather than trying to be upright. So that's where the bear walking and the crab walking, which is basically on your all four limbs, either forward or backwards, and working on the core, which would be a really good idea. So exercises that will help with the core. Swimming helps with the core if you do have connection to a pool or water, as well as if you do have an ability to play those interactive sports. Anything, and if you don't have those interactive PlayStation games, that type of thing, even just having some fun with being on your high knees, I'm going to do this here, and having somebody just playing a little bit of like karate moves, you can always have some fun doing that. That does help with the core strength. And again, we always make things into a game in our house, so we always try to do that. And having, if you have access to a physiotherapist, that's probably the best, thing that would be good, and having a lot of suggestions through that, because again, every situation is unique. And if there is any balance issues, then you do have to do as much as you can to protect the fall.
Then we can share the microphone and we can do the questions in the chat. Would that be good? A question.
Absolutely. I will pull the page. Question 1. Is 1 guanabens, which is used in vanishing white matter, possibly effective for MLC?
No, I don't think so. It has nothing to do. So that's specific for vanishing white matter. That's a short answer.
Just going to post that for Italian and Chinese. I was supposed to question. Different on the answers.
If you give me a new question,
I'm going to give you a new question.
then you can type.
Perfect. Question 2. Do you have any information or opinion on the use of anakinra, also known as Kinred, on MLC?
Thank you for the question. That's a very good question, as our Italian collaborators showed, and I had it on the slide also, that there was something also involving interleukins in MLC and inflammation, and then you would think that anakinra could be an option. The thing is that I think that good options should always be tested in reality and not just be tried out. So what we are going to do, we already contacted the group in Rome, and they are great in inflammation, and we have an animal model that we are going to try anakinra in the MLC mice and do a collaborative study with Rome to see whether there's any effect, to give it a basis. And then if it has a basis, then I still think that we also should have an, even if it would be a simple trial in MLC patients, because MLC patients are not the same as MLC mice. So there's always this translation. And so I think we should find out whether that's a good idea. But potentially that's an idea, yeah.
Are there methods that can reduce autistic or behavioral problems in MLC?
Thank you for the question. That's addressing a realistic problem in MLC, that some patients have autism or behavioral problems. And personally, but I'm happy to hear what you have to say, Jigyasha. The approach is the same as all patients with autism or behavioral problems. So it's not that you need to adapt the therapy, the psychological therapy, to MLC. So you can just, in the Netherlands, I would contact an autism team and then ask them to accept this patient for treatment.
Question 4. Can muscle lengthening or muscle transfer benefit MLC patients with spasticity?
That's also a great question. And I think that by lengthening and transfer, surgical procedures are meant. And I would say spasticity is a symptom, and independent on whether it's caused by, perinatal asphyxia or whether it's caused by metachromatic leukodystrophy, it needs to be treated. So I come from, I'm in the field for a very long time, and I come from an area, from a time, an era, I should say, in which some colleagues thought that you should not treat spasticity in metachromatic leukodystrophy or MLC because it would get worse and why bother. But since we are much more aggressive on treating spasticity also in leukodystrophies, we see the benefit for the quality of life. Patients are doing much better, so their life becomes much better. So the answer is fundamentally yes.
Some families are using a vitamin called Neural Optimizer, OPTIMIZER. Do you recommend it, or is it necessary?
It sounds like I need to take it myself. I would love to take it. But the answer is no. I don't think that, I think that if you listen to the story of, for instance, Rogier and Quinty, that there is no simple drug treatment available, and it has nothing to do with enhancing vitamins or radical oxygen scavengers or, so the answer is no. Although I'm tempted to take it. Not beneficial?
Sounds great. Question 6. In regards to research development, can CRISPR-Cas9 genome editing possibly repair the DNA that it's involved in the MLC gene defect?
Yeah, of course, also a great question. It's a monogenic disease. That means there's one gene that needs to be fixed. So fundamentally, the answer is that gene editing is a possibility. CRISPR-Cas in vivo is still not safe because CRISPR-Cas editing also may make new mistakes. And I think gene therapy right now is still sort of something that we need to work on because it's fundamentally a good idea, but it's not ready yet. And for CRISPR-Cas, I think there will be quite some years, if ever, and there might be other ways of gene therapy that will be applicable. I spoke about this with our colleague from Rome, Dr. Ambrosini, and she shares the idea that if we are able to develop drug therapy that would be easier and preferable regarding also side effects. What I find important to realize that everybody should realize, of course, the end outcome for MLC is not good, but as a disease, it's very slow. And what I feel is if you cause damage, then that might be acceptable for a very bad disease like spinal muscular atrophy type 1. Patients typically die in 2 years. So, but if you have a very slow disease and you cause damage, then as a doctor, I find that unforgivable. And so that's something that I think we need to balance the risk versus the chance of risk versus the chance of benefit.
Question 7. Is the surgery that is used successfully in cerebral palsy possibly helpful in MLC?
Yeah, it depends on what the problem is. But indeed, if the problem is serious spasticity, then the assessment is the same. And at least I would feel that placement of an intrathecal baclofen pump for a child with severe spasticity is equally indicated if it were another cause. So the answer is that it needs to be assessed what's good for this child, and then it needs to be done. I see all sorts of characters popping up here. Yeah. It's amazing, huh, that I master all those languages. I also didn't know that until today.
Question 8. Dr. Mario, do you have any information about the treatment studies that are going on in Turkey? Are you in contact with them?
I'm in contact with mostly with Dr. Pinar Topologlu. She worked with us in Amsterdam for quite a while, so we have great contact. Just like also Dr. Sinha, who was in Amsterdam for a while, and so if there are studies, I know that some doctors in Turkey gave anakinra to children outside of a study, and I'm sort of not very happy with that, because then you really don't know whether it helps or not, or doesn't help. I think that experimental treatment should be in the context of a formal trial, because otherwise there will be no data on effectiveness. But it should be clear that we founded this consortium of clinical MLC experts also to form an opinion on such questions. It's an amazing system.
Pretty crazy, Dr. Sinha, this question is probably directed at you. I wonder how many MLC patients are registered in India. Does MLC1 progress with a severe course in individuals in India, just like it does in Turkey? Do you see similarities in the severity of the phenotype?
Yeah, thank you very much for that question. Again, very interesting question. As I mentioned that we do not have a registry as of now, but we are planning to start that very soon, because each of the doctors in India are having individual database. So we are going to pull that up and have a registry to start with. The second part of the question is about, again, the phenotype, I suppose. Yeah, so yes, again, we do not have scientific data at the moment about the phenotypic variation, but we have observations that there is phenotypic severity variation with the same genotype. But we really need to look into that in due course of time. So MLC1 has different severe progressive differentiation is there. Someone can be severe, someone can be mild, but we have to look into that. Thank you.
sorry, I think there's some new questions. Have you been able to follow them at all? Lovely. Give me a second, sorry.
I don't sing well.
Do you think it would be beneficial for the families and patients in India to create like a kind of brochure about the disease? Do you think that would be something that they'd be interested in getting, and do you have plans for that?
Thank you, Alex, for that question. It is indeed a very important, pertinent thing for us because, yes, I am planning for a disease bulletin. I am going to publish that and I'm going to share with my patients as well, and I'm going to distribute that among the neurological societies of India. The reason is that there are many who are not that technologically savvy, who can, access the internet that well. So at least the disease information bulletin can help them. That is why I mentioned the organization of Agarwal community also, where I can hold certain meetings, maybe once a year or something, and also, give them the disease bulletin so that awareness can be created through that as well, and some updates on the disease as well. So, thank you.
Question. It is said that when the treatment is found, there will be a complete recovery and he will be a normal, healthy child. Do you have a comment on this?
Okay, thank you for the question. I think that's an important question because I'm all for clarity and I'm against false hope. The problem with the brain is that it's not good in repair. So if somebody had a traffic accident and bad brain damage, then after, let's say, a year, you know how much the recovery is, and then after that, it's stable, and it's not that it's the brain is growing back or something. So with leukodystrophies in general, you can say that what we are aiming for is to reduce or stop disease progression. And if you get anything back, that would be a bonus. But that also means that if you can start the treatment early, that the outcome might be that the child has no disability. But that means that for the children who are already there, and for instance wheelchair dependent, I'm very doubtful on whether any treatment will get the function back. Dr. Sinha, do you have anything to say about that?
No, Mario, I think at the moment we are looking for, slowing the disease progression is what you already mentioned, and I think that is the most important thing as of now. Yeah.
I think if you raise your hope too high and you think of something, there will be a miracle treatment that will completely make the disease go away and lead to a total recovery, that's setting yourself up for disappointment. And I always say to my patients, disappointment is really a bad emotion because that makes you, sad, angry. So I think you better be realistic. Marla, is there anything you would like to say about this? No? Yeah. Yeah. And the positivity is not only in the ability to walk. A lot of people that I speak in my practice is their focus is mostly, is my child going to walk again? And then I think, but I think in fact the most important capability of a child is to communicate. Is to be able to make clear what's ailing, what he wants, what the wishes are, what the sadness is about. That's actually by far the most important capability. Marla, you want to say something?
I will just be a moment. I know that many people that know our family and have told, let's say, Ron's mother, so their grandmother, not that this is hopefully going to sound bad for Alex and Liam, but that they said of the 3 kids, that Aiden has always appeared to be the happiest of the 3 of them. So, I mean, and Liam and Alex are very happy. However, so when people would see somebody who is sitting in a wheelchair, they would assume the opposite, right? But this just goes to show you that there's more beyond what you see, and that, you know, you have to look at the optimism of everything, and that just because somebody can't do things what we perceive as normal or natural doesn't mean that you cannot live a very happy, healthy life. Okay? I just wanted to share that.
I think that's a very important thing that you just said. And I think it's also, I feel like it's also judgmental. If you judge a person by the motor capabilities, and you know, and Aiden is in a wheelchair and his mother is behind it, and you speak to his mother, that's judging as if he's mentally deficient. Assuming that he's unhappy is also judgmental, and it's narrow-mindedness. That's what I feel. You have to be more open and think that there are other ways of being happy.
No. I have another question. It's about epilepsy specifically. My son has epilepsy. We have finished with the treatment. He's currently using, I believe it's only Depakote. Will it trigger the seizures once the treatment is over? And I would imagine that would apply to all medicines, not just Depakote.
Thank you so much for the question, because it's not that Depakote or any other anti-epileptic drug cures the epilepsy, so that there will be, if you take it for 6 months, you're done with the epilepsy. So if the child has epilepsy, then it's a good thing to continue it. And if the epilepsy maybe, you know, sometimes after 2 years of not having had a seizure, the doctor may try whether you can now live without the drug, but it may come back and then you need it again. It's fundamentally not curing the epilepsy, it's suppressing the epilepsy.
Another question, once again, epilepsy specific. Is it required to use epilepsy medicines because the EEG image is damaged in an MLC patient without a visible seizure? In other words, do you need to use epileptic medications because their EEG's off, but they've not had seizures?
No. I always say that EEG is worse than the patient, because on EEG you see more epileptic activity than you see in real life. And if there's clinically on the outside no epilepsy, then I would not start anti-epileptic drugs, because they also have a general effect, like they slightly decrease cognitive abilities, so the school performances are going down. So you really need to use it for something that's clinically relevant, and not just to suppress EEG activity.
Perfect. I've come to the, I believe, the end of the list of questions. If I haven't addressed something, please throw it in the chat now, or of course, live from the audience. But I don't see anything here.
Yeah. I'm going to give you the microphone so that it can be all in the open.
So my question is, do you see any indication of a direction of possible brain-machine interface or neuroscience utilizing technology for increasing movement or bringing movement function back through machines and technology?
Thank you. While I was listening to you, I was just looking at the slide and I thought that somebody put it up for me. Anything is possible. I would not say yes or no. I just don't know. I know that, for instance, that for blind people there is technology in development that an external sort of machine can take over and then bring it into the brain so that the person can still see. So I'm not sure what the future holds. Technology is also developing. Is there anything you would like to say about it, Rogier?
I think that what you say is right, that anything is possible, but because similar for the blind people, people are also working on things like that for people with paralysis, and I must say that I've never contemplated that in the context of a disease with spasticity or so, but I think it is imaginable that it would work. I would not be able to say how far off that is, because technology moves in such strange speeds. So I think it's a very interesting question. I'm not sure whether people are pursuing it. Yeah.
I think what is against MLC is that diffuse disease. Like if you have a traumatic transaction of your spinal cord, I think it's close by that indeed with technology, the message from the brain, I want to walk, is transferred over the lesion into the spinal cord so that people can walk. I think that's half in practice, I would say. The thing is that we have no specific lesion to bridge in MLC. That's it. The problem is everywhere. So I think that those diseases are more difficult to deal with. But I'm not going to say no, because when I first I was on an ERC committee and read the proposal for blind people, and then I was just amazed by what technology can do, and surprised because I didn't know about it. So I'm not going to say no. I'm going to point to Marla's wise words. Anything is possible, but not right now.
A new question. Is there an MLC center that you know of anywhere in the world where a family could go and see all the related doctors you might need?
That's also a good question. There are very few centers right now that are clinically focusing on MLC. To be honest, we haven't set it up specifically for MLC in Amsterdam, although we do have leukodystrophy clinic, and people can come see us, and we have all facilities. And I think that Dr. Sinha Jigyasha is going to set it up specifically for MLC, but she also has the numbers. I would not have the numbers in the Netherlands for having the justification for an MLC clinic. Well, perhaps I should add what the good thing about this time is that we are so connected worldwide with each other. So the knowledge that's generated on the subject of MLC is, as soon as we publish it, it's on PubMed and it's available for everybody. So lack of knowledge is actually not justified. Any other question?
Are there any diseases that are similar to MLC that are equally rare, that the scientific community could join hands with?
I have to think about it. I do think we do look at other diseases because MLC is related to several other disorders. Like also in some congenital muscular dystrophies, there are very similar white matter abnormalities, and we are looking at it. So we are certainly also looking over the hedge into other gardens to see whether there's anything, any flower we can use. And if we can join forces, we'll certainly do that. I think our first duty is, and I'm glad that we have this meeting in February, is to join forces within the MLC field, because I think with a disease that's so rare as MLC that it's easily getting scattered. You know, every doctor sees 1 or 2 patients, and then the patients don't communicate with each other, and so I'm very happy that Alliance MLC has been formed, and I'm proud of the MLC consortium, and we'll see what comes out of it.
The question is, what's the time frame for a possible outcome, which I guess would probably mean a possible treatment? Yeah.
I would not expect something within 5 years. I think that's fair to say, because I don't want people to feel like it's possibly they find something next year. If I see how it goes with other disorders, it's a long way to go. But if we are discouraged by the length of the way and don't start at all to walk, then we'll never get there. So we have to just to walk the way, and then we'll be surprised by the time it takes. It might be less, it might be more, but we need to get ready and need to start walking.
With the recent advances on mRNA, are there any researches in the matter of producing the necessary proteins without depending on DNA that could be used for some sort of gene therapy treatment?
No, I just think that's also something, like I said, about gene therapy. It's just not ready yet. That's really something that needs to be worked on. That's certainly not going to be ready anytime very soon.
I think we're running fairly short on time, so why don't I continue to monitor the questions here, and maybe I'll put them into a document and email it to you, and then we'll post them on the Facebook group perhaps. Does that make sense? Yeah. Wonderful.
I just want to thank everybody for being here and for joining the meeting online. Thank you very much for being there, and also for contributing with important questions. And I thank the speakers for contributing important talks. Thank you so much, and have a good day.